Just a brief overview. As mentioned in the invitation, this is a brief overview about the microRNAs, the history and the current status, then how it relates to the endocrinological field. I'll mention a recent review paper, and then highlight two of the most popular methods which are used for discovery as well as profiling, which are microarrays and next-gen sequencing. And then we will finish up with a few case studies. Some of them are customer publications, some are not. And while I won't have time to go into detail of every paper, of course, due to time, I hope it will give you some inspiration and some ideas what you can do in this field when it comes to microRNA research. And as mentioned, if you have any questions, type them in before, or of course afterwards we will stay on to make sure that we cover everything. If you're new to the microRNA field, chances are that you haven't heard of LC Sciences before. We are located in Houston, and so far rather microRNA-centric company. We exist since 2004, 2005, and we were actually the first microRNA microarray service provider. The core technology that this company was founded on is a microfluidic chip technology, which allows us to provide services for pretty much any type of microarray. We can customize this, and so this is why the founders back in 2005, when this company was founded, had the idea to, or the vision, I should say, to jump into the microRNA field, which was just evolving back then. Our customer base is really worldwide. More than 40 countries is what we currently cover. A lot of academic institutions, of course, but also an increasing number of biotech companies as well as government agencies like, let's say, USDA or FDA. As mentioned, the core technology is really our microfluidic chip technology. In the meantime, we added on sequencing services as well as qPCR validation using the TechMan system. So that's really, the TechMan system, of course, is not ours. We operate with a license there. It's just as a convenience for customers who want to validate array data. Besides microRNA arrays, we also offer proteomics arrays. But so far, really, the microRNA field has kept us so busy that this is still the majority of our focus. So let's start with what we know so far about microRNAs. They are small non-coding RNAs that are usually 20 to 22 nucleotides long. This is the case for animals. For plants, they're typically a little bit longer, up to 24 nucleotides. The mature microRNA, this is typically what people mean when they talk about microRNAs, are processed or derived from a so-called precursor molecule, which is a rather stable folded molecule which has a stem loop, which has a stem structure as well as a hairpin loop structure. And as mentioned, that's very, very stable. Typically, it doesn't unfold during hype conditions of a microarray. So typically you don't detect that. microRNAs are evolutionarily very highly conserved. So there's a number of microRNAs that are identical in sequence between worm and human, for example. And of course the same is true for the plant kingdom. The main function, or the function of microRNAs, is that they bind to messenger RNA molecules and act as negative regulators of the translation. And this can happen in two different ways, which I will mention in one of the following slides. In order to work or to function as this negative regulator, they have to be present in a really high copy number, which is the case. And what makes them very valuable for diagnostic purposes is the fact that their expression is tissue and developmental stage specific, and of course also disease stage specific. Currently, there are more than 25,000 mature microRNAs known. They derive from roughly 21,000 precursor molecules, and all these sequences are kept maintained in NearBase, which is the public database. It's currently hosted at the University of Manchester in the UK, and it covers 193 plant, animal, and virus species. The number of updates kind of slowed down, but the last update was just roughly 6 weeks ago, beginning of August of this year. The previous update before that was in November of 2011. These are typically all known mature microRNAs or known precursor molecules that have been experimentally verified. So these are not just predictions. The mechanism of microRNAs is somewhat understood. It's rather complex and far-reaching. This is due to the fact that each microRNA can control multiple genes. Last week I read an estimate that probably each microRNA can target up to 100, if not 200, different genes. And from what's known about the human model so far is that roughly 1/3 of all human genes will be regulated or are regulated by microRNAs. It's a negative regulator of translation, as I mentioned, and this can happen in two different ways. The hypothesis so far is that for plants, predominantly plants, you have complete pairing, meaning that the microRNA sequence is the exact complementary of the messenger RNA that it binds to. Of course, this is a very tight interaction and results, well, the result of this is that it triggers the degradation process of the messenger RNA. In the animal model, it's more prevalent is the imperfect pairing, so that you have quite some mismatches allowed between the microRNA sequence and the targeted gene. So the interaction is not that strong. Typically, the degradation doesn't get triggered, but it inhibits the translation. When you look at the milestones, it's a really still, even in 2012, is still a pretty new field. Just the speed of publications and so on really started to pick up in the last couple of years. But it all started in 1993 when Lin4 was shown to encode two small RNA molecules that actually, so the unique thing was that it encoded small RNA molecules and not proteins. So it was kind of very unusual and got dismissed by many. And then in '98, it continued with, then for the first time ever, RNAi was observed, and it resulted in the Nobel Prize in 2006. So these discoveries of these small RNA molecules that somehow had a regulatory effect was, of course, a major challenge to one of the central dogmas of biology, which assumed that DNA gets transcribed into RNA, and RNA then translates into protein. So the small molecule that had a regulatory effect and didn't really translate into a protein was not on the map back then. Then in 2000, LET7 was identified in humans and in Drosophila. And then from then on, everything picked up in speed. So 2001, Bartel, Tuchel, and Ambros together discovered basically the large class of small regulatory RNAs that we call microRNAs today. Then didn't take very long, and already in 2002, the first hints about the processing were revealed. The first piece of the puzzle that was found was basically DROSHA, which is processing the primary microRNA into the precursor microRNA. Then in 2004, microarrays were first used to profile the microRNA expression. And then 2005, next-gen sequencing was already adopted to the use for small RNA discovery as well as analysis. Then in 2006, MirBase, the first version of the public database, went online. It was founded earlier, but that's when it went online. And initially hosted by the Sanger Institute, now in Manchester. And in 2008, circulating microRNAs were first described and detected in body fluids. In the meantime, microRNAs are known to be in a whole variety of body fluids, starting, of course, with blood, serum and plasma, of course, and as well as urine and even breast milk. When it comes to endocrinology and how microRNA research relates to it, there are a couple of milestones, actually not that many, that we put up here. Basically, 2003, it was discovered that microRNAs play a role in the energy metabolism, specifically microRNA 14, which suppresses cell death and is required for the fat metabolism. And 2004, microRNAs were shown to regulate a whole variety of endocrine functions, from insulin secretion, adipocyte differentiation, and as well as B cell development. In 2007, microRNAs were first described to play a role in diabetes, and in 2008, then microRNAs were linked with first linked with hormone-dependent cancers. Of course, microRNAs were linked to cancer much earlier than this, but that's this is specifically for hormone-dependent cancers. And if you look in general at the different other fields, let's say cardiovascular field or cancer research, we feel that the endocrinology field still has there's still lots of potential and lots of research that can be done in this field. It's still not completely untouched, but still a lot of white spots on the map. Just to give you an overview briefly about the really rough outline about the microRNA processing, the gene that gets translated first forms the translated product is a primary microRNA transcript, or prime microRNA. It has, as mentioned, the stem region as well as the hairpin structure. And this little bulge here is already indicating that these the stem region is not a perfect complement. So there are mismatches that you will find. There's at the 5' end there's a cap, as well as on the 3' end there's a poly A tail at this point. And this molecule then gets processed by Drosha and Pasha to protein complexes into the so-called precursor microRNA. What's happening here is basically that the 5' cap and the poly A tail are being removed. This all happens in the nucleus. In the case of plants, the further processing happens in the nucleus, but in the animal model, it then gets transported into the cytosol, and this precursor microRNA, which, as I mentioned, is rather stable and doesn't unfold very easily, is being processed by a DISA molecule into the microRNA star duplex, how it was described back then. Today we would say it's the resulting duplex is the microRNA-3P and -5P duplex. So what's happening here is basically the remaining ends are being cleaved off here at the 5' and 3' end, as well as the hairpin loop gets removed. The reason why I mentioned that nowadays we would call it differently is because as of the last MirBase update, so since 6 weeks, for all species, a new nomenclature has been enforced or applied, I should say. In the past, and what you will find in the literature, you will oftentimes see microRNA with the asterisk. That used to be the antisense, or the so-called minor, but it was never really clear which one was which, so now it's much easier and much more uniform. Each microRNA has now a -3p and -5' naming to it. So this duplex can then dissociate, and the resulting product is what we call the mature microRNA, which then can bind to messenger RNA and form the miRNA-seq complex, which is the microRNA-induced silencing complex. For the latest about the nomenclature, I can highly recommend the mirbase blog. It's mirbase.org, and then there's a link to that blog. Sam, who's maintaining mirbase, he does an excellent job in explaining the changes and the reasoning behind it. If you look at the mirbase content over the last couple of years, this is basically since showing the number of microRNAs in the database. These are the precursor molecules here that are listed. And that's something that's kind of confusing at times. There are different numbers thrown around, and sometimes people talk about the number of precursors and sometimes about the mature microRNAs. So that's why you see sometimes different numbers. So in this case, these are the precursor numbers, and shown from version 6 all the way to August, so last month, to version 19, and you can see this almost exponential increase in sequence information that's available. And hand in hand with that goes, of course, the almost exponential growth in the number of microRNA-related publications in PubMed. And from almost nothing in 2001 to probably way over 6,000 this year. So besides the fact that microRNAs seem to be really interesting, why should you study them in the context of endocrinology? There are a couple of reasons, of course, and some of them are identical to other research fields, but it's clear that there's a relationship between these two fields that has been described as intricate reciprocal, meaning between the hormonal system and the microRNA regulation system. Many microRNAs regulate hormones, and on the other hand, many microRNAs are regulated by hormones. And microRNAs have shown to target many genes that are important for the proper endocrine functions and metabolism. And of course, last, dysregulation of microRNAs can contribute to endocrine-related diseases. Hormone-dependent cancers, of course, is a big one. Obesity is becoming more and more clear during the last couple of years. Then diabetes, we've already mentioned, hyperglycemia, as well as lipodystrophy. So if you look at the clinical potential that microRNAs might have in this field, or in the meantime, it's clear that they certainly have this potential. It's the first thing, and of course the low-hanging fruit is always the use as biomarker. Basically, you're looking at the microRNA profiles between different groups, different sample groups, control versus disease, and see which particular microRNAs are up and down regulated. The samples that are being used is, of course, body fluids, more recently, as a non-invasive way of testing, in particular, of course, plasma, exosome, as well as HDL particles. And it has certainly the potential to make accurate and detailed clinical diagnosis, especially in the cancer field. And what's mentioned here at the very bottom is the first diagnostic test that's commercially available already, made by Rosetta Genomics. It's called MirView, and it's able to differentiate between 4 different types of lung cancers. And this is, of course, very important because it's a non-invasive way if you use blood. And typically, the clinician has a hard time differentiating between the different lung cancer types. And it's important because, of course, the the treatment has to be adjusted accordingly. And so far, the accuracy of the diagnosis was without this test was around 50 to maybe 60%, if you were lucky, and now it went up to way above 80%. Another potential use is the use as basically to determine the prognosis and predict treatment efficacy, as well as to monitor the effects and also to predict maybe the trying to assess the molecular effects of environmental toxins and other environmental factors. In this context, I should mention so-called endocrine disrupting compounds that one of the papers I will mention is focusing on. So initially here, people thought that it might be enough to just look at one particular microRNA, but it turns out that you actually have to look at the whole set of microRNA signature, which typically consists out of 15 to 20 different microRNAs. Moving on from up from the purely diagnostic field, of course, microRNAs are also a potential drug target, either by inhibiting them or by reinstating their expression levels. So in the later case, of course, then as a therapeutic. So in other fields, especially the cardiovascular field, a couple of companies were founded already in 2006-7, around that time, and they are making already progress in developing drugs that are relating to the cardiovascular field. So, and when you study these microRNAs and look at the differential profile, of course, it also helps to give insight into the mechanism of infection, stress, or other stimuli. The review paper that I wanted to mention is focusing, of course, on this field here, and it just came out in April in Nature. Nature Reviews. It's titled "MicroRNAs and Metabolism and Metabolic Disorders," and it's a very comprehensive overview, as in the review should be, of course, about all microRNAs that are known to be involved in the different metabolic pathways. So it highlights microRNAs as key regulators of cholesterol, as well as lipid homeostasis. Their involvement in the insulin signaling, and basically all other endocrine-related pathways, including obesity. It also lists the particular microRNAs that are involved and in which pathways they are involved. So this is the table from the paper. As mentioned, you will get the PDF file with all the links in here. So this is a whole long list of microRNAs that list then here in the next columns at the targets, the potential function, and the verified or predicted target genes. So this is still clearly the bottleneck, because each microRNA can target multiple genes. The information about which exact gene is the actual target of a microRNA is still rather limited, especially because it doesn't have to be a perfect match between the sequences, because since mismatches are allowed, that makes it even more complicated. So this just as a brief overview, you see the numbers of microRNA numbers are rather low. The numbers that I'm talking about are here, for example, MIR29 or MIR32EA. These are really low numbers, and it means that these are microRNAs that were discovered early on. Basically, the earlier the microRNA was discovered, the lower the number. Another review that was also published around the same time this year in May is not really specific to the endocrinology field, but it's extremely helpful in my opinion. It's basically a review paper about the different profiling methods and what you should consider with each method, and also depending on the sample type that you have. So what you have here is, I took out two of the graphs, but the paper is really very, it highlights every single method, mostly of course focusing on qPCR, microarrays, and rna-seq, and it does an excellent job in really highlighting the pro and cons of each method depending on the sample type. And so this is what's shown here. You start out with, depending on which sample type you have and what type of total RNA harvesting or sample treatment you're applying, which method you should choose, and also this flowchart here, it helps you to guide the right method depending on the question that you're trying to ask. Because this is something that we see all the time, is that people tend to believe the latest and newest method or try to apply it even though it might not really be the right method to answer the question of the experiment. So this is really an excellent paper in that regard. So when we look at the tools that are available for the different steps, of course everything starts with the identification of microRNA. In the early days, there were a lot of tiling arrays that were also performed here by customers, and one of the earlier mirror-based updates pretty much exclusively relied on data that came from our customers here. And nowadays, of course, next-gen sequencing is the clear method of choice for this step. But other methods include genetic screening, then cloning strategies, of course, as well as computational methods, especially if in the plant field, etc., there were a lot of computational strategies applied. And if you move on from the pure discovery and want to do the actual detection and profiling to see whether you have any differential expression between groups, of course you can use northern blotting, which was used quite a bit, and in situ hybridization, and then real-time PCR is probably one of the more popular ones. What I will focus on in the following are, as mentioned before, the microarray analysis method, as well as next-gen sequencing. And then we move on to the target determination. That's, like I said, still there's still a lot of white spots on the map. Method of choice for many is simply to try to apply bioinformatic tools like TargetScan, Pictar, and Peter. There's a whole variety of databases out there that's trying to determine this. But like I said, it's not that easy because mismatches are allowed, and each microRNA can target multiple genes, and this the target might depend also on the developmental stage as well as the disease stage. So it's rather tricky. So don't be surprised if you enter the same data in any of these databases or these tools and you get completely different results. Then of course the goal will be to do really experimental validation of the target determination, so using pull-down assays, 5' arrays analysis, or in the plant field really big coming right now is degadome sequencing. When it comes to the pathway analysis, there are a couple of useful tools out there, but again, that's also still, there's lots of room for improvement. Some free sites or databases that try to bundle the information that's out there are MirFocus, MirPath, and MMIA. There are also commercial tools available in the meantime. They pretty much do the same thing. It's just that they are being curated on a more regular basis, whereas the free tools typically rely on other free databases that are not maintained as frequently or not updated as frequently as the commercial tools. Then when it comes to functional analysis, you can use luciferase assays, knockout models, of course, that's probably the most common one, as well as trying to inhibit your microRNA with antagomeres, or the other way around, try to see what happens if you add a microRNA mimicry to reinstate expression levels of a particular microRNA. As mentioned, I want to focus on the profiling step and the discovery step using the two different methods. The typical workflow used to be for years that you do an array, and of course that's also true for the gene expression field. You would do a microRNA microarray and then validate these results with qPCR. This had, of course, the advantage of being able to scale to screen a big number of microRNAs on the array cost effectively, and then get the quantitative measurements using qPCR. And with the rise of next-gen sequencing, of course, this is now the clear favorite step for discovery. And what turns out to be a very powerful combination is really if you combine this next-gen sequencing step with the then custom microRNA arrays that are designed specifically based on the next-gen sequencing data. And this is something that is now standard for a lot of customers of ours, especially when you're dealing with species that are not well characterized. And we call this basically seq array. The way it works is that you sequence only a very limited amount of samples, often only one pooled sample, and by pooled I mean we pool in this case different developmental stages or different disease stages, so that we cover the entire potential range that might be present in the samples of the experiment. And we sequence this and create the custom layout for the specific species as well as experiment for the microRNA arrays, and do then the actual profiling with a large number of samples using the microarrays. This way you combine basically the power of the discovery with the cost effectiveness of the arrays. Before you can start out your experiment, and no matter which method you use, of course, sample preparation is key. In the meantime, there are a lot of different commercial kits out there. Here are just 3 examples. Norgem Biotech, they have an excellent RNA extraction kit, and they have also a lot of specialized kits depending on the sample type and also the volume that you're dealing with. QIAGEN has a particular microRNA kit which is called rna-seq kit. With QIAGEN's kit, you just have to watch out for make sure that you're picking the right one, because they have another kit. It's called just rna-seq kit, and it's also described as total RNA extraction kit, but it was developed before microRNAs were known, and it doesn't retain microRNAs. So either you have to modify the protocol, or the safer route, of course, is you pick the right kit from the start. So this would be the mRNA-seq kit, and Ambion has also an excellent kit. It's called Nirvana. No matter which kit you pick, make sure that you stick to it throughout the experiment, because while they all work, they will introduce a slight bias in the profile. So each profile looks a little bit different. So you want to avoid that additional variation here in your experiment. Of course, a lot of people are also using the old-fashioned trisol method, and you can get excellent results with it. Our observation is that especially people that are new to the field and are not used to working with RNA or microRNAs in particular, and they really struggle to get good sample quality. There are lots of degradation issues typically. So people that are familiar with Trizol, they of course, and if they had good results, of course, feel free to use it. Again, the Trizol method was of course developed long before microRNAs were known. So you have to make sure that your protocol is really also extracting the microRNAs. You have to increase the isopropanol concentration here, and we have a modified protocol on our website under the frequently asked questions. When it comes to sample types, of course, you have the whole variety. Tissue samples usually give you really excellent results. Then, of course, cell lines are very common also. Then in the clinical field, FFPE samples are common as well. The sample quality here is less problematic than for microRNAs as compared to messenger RNAs, because microRNAs are already so short they typically don't really get fragmented during the initial preparation. But the sample quality really depends not so much on age, but really on how the initial sample, how the paraffin was added, and so on. So that is, if you inherited a collection of FFPE sample, that's really out of your control. So typically we run a few samples of each collection first to see how the sample quality is. Then nowadays, of course, more and more are blood, serum, and plasma samples that are becoming more popular because it's less invasive, and then as well as urine samples these days. The sample amount, typically we ask for more, but we can deal with 500 nanogram. Typically the default amount that we would use is 1 microgram of total RNA, and we would ask you to send 2 microgram. So no matter which kit and method you're using, make sure that it's appropriate for your sample. All these kits mentioned here, they have come with a whole variety of different protocols depending on the sample type that you're starting out with. If you want to check the quality, you can look at the UV spectrum of your sample. What we look at is with a nanodrop is a ratio of 260 to 230, which should be above 1, and the ratio of 260 to 280, which should be above 1.8. What is shown here on the right is an Agilent bioanalyzer run. We don't routinely use that simply because the prediction about or the statement about the microRNA quality is rather limited, but of course it gives you a great idea about the overall RNA, total RNA quality. But we had really cases where we had RIN values of 10, but the microRNA was completely degraded. So this little hump here is the microRNA, and these are the 18 and 28S peaks. So this is not something we routinely do, but on a case-to-case basis. Of course, the best method is still to run a gel, but that's not really high throughput, so we typically don't do this here. These are just some pictures to give you an idea how it should and should not look like it. These are courtesy of Nordgen Biotech here. These are different types of gels, formaldehyde, bioanalyzer, and urea page gels, and you have always good total RNA extractions where the small RNA here down here is retained, whereas in the poor samples these samples show hardly any small RNA, so they weren't really not retained. Or the other option, of course, would be that they're completely degraded. Besides the extraction method, one really important aspect is the number of biological replicates. There's certainly the trend that people tend to think that with more advanced analytical methods or discovery tools like next-gen sequencing, that it somehow will work with less biological repeats. Of course, that's not true. The statistics are pretty much the same for no matter which method you're using. So the minimum number of repeats is still 3 to do any type of real statistics. If people send in just one sample each, then of course we can give you can state you the difference between these two samples, but of course there's no way of telling what this is of any biological or statistical significance. This might be just some random variation that you're observing, and you will never know. Same is true when you pool samples. If you send us only one pooled sample, of course there's let's say one pooled control and one pooled disease sample. There's still no way of doing any statistics. And if you have one bad sample in there, that might skew the entire result anyway. So no matter which method you're using, use a reasonable amount of biological replicates. So for example, if you have the choice between doing next-gen sequencing with just 2 samples, you might want to reconsider, because for the same price you could get easily a microarray performed with 6 samples. The number of replicates, of course, depends on the sample as well as experiment type. So if you're working with with animal tissues, that's what I was just referring to in terms of 3 replicates. If you work with actual patient samples or clinical samples, then you might want to use at least 10. And another important factor, of course, is also which paper you are trying to get into, and the reviewers oftentimes demand 30 or more replicates in that area. Another point is to make sure that the number of samples in each sample group is roughly the same. So it doesn't make any sense to let's say use 10 treated versus 1 control sample. Or even if you have 2 control samples, it's still heavily biased. So ideally you have the same number of samples across each sample group. If we briefly look at the advantages and the features of both methods, array versus sequencing, if we focus first on the array, of course it's a proven method. It's rather robust. It's fairly high throughput, and the data that you're getting is relatively easy to analyze and to comprehend. It's low cost. Of course, you have to know the sequences already. Well, we can synthesize custom chips with predicted sequences or so. That's not a problem, but in general, you have to have, of course, some kind of probe content. In our case, we synthesize custom to order and basically look at what is available in MirBase that day. So the default content in our case is the content of MirBase. So this is a really great method if you are working with a well-characterized species such as human or mouse or rat or similar. And this is just a brief overview of our chip technology. That's our smallest chip. In the meantime, we're also using a larger one. This one has 4,000 features, and you see these are the reaction chambers, so they are not exactly round, so that's why our scans also look a little unusual. But basically, in each of these reaction chambers, we can synthesize a different sequence using photochemistry. And this happens, as I mentioned, custom to order. We start the synthesis of the chip for a particular experiment once we receive the samples. And this allows us really to keep up with the constant increase in sequence information in MirBase, which we call MirBase synchronicity. What are listed here are the number of precursor sequences again from MirBase version 1 in 2003 to all the way should change that to 2012 here, with version 19. So this is the latest version that we offer. It takes us typically a day or two to adapt to new probe content, but that's really it. If you look at these all these numbers, there are two main incision points to mention. One is the switch from version 10 to 10.1. It doesn't look like a big jump here in the number of information and the number of sequences, but what happened here is that a lot of microRNAs were renamed or sequences were corrected. So if you're comparing, let's say, microRNA number 10 with version 10 versus microRNA number 10 with a mere-base content 10.1, it might be not the same sequence. So because this is a continuing problem in the microRNA research, that the names keep changing, and also the nomenclature, as mentioned, it's always the safest to really compare based on sequence. The other important incision point here was in last November with the release of version 18. The new nomenclature that I already mentioned was introduced initially only for a couple of species, human, mouse, I believe zebrafish was also part of this. But now since August, so since last month, this nomenclature change was applied to all species that are listed in mere-base. So again, if you want to compare data here, it's becoming a little tricky. I would say right now the majority of the arrays and even qPCR primers and so on, they are now still referring to the old nomenclature, so all of that has to be updated. And yeah, as mentioned, we offer already version 19. It's just a quick workflow. What happens once we receive samples. I won't really go into detail here, but basically we do sample QC, labeling, hype, do the image scan, data extraction, data analysis, and send back the report. And typical experiment, which includes multiple biological repeats, looks as following, or the results that you're getting look as following. You have multiple biological repeats, you do the arrays, or we do them, and then after normalization, we generate this heat map here, which includes all microRNAs, all samples, and it's always fascinating to see how disorganized and chaotic this oftentimes look. And then just by applying a few simple statistical methods like t-test or ANOVA, you can really differentiate between these sample groups. And what you're getting then are here two examples of heat maps at different p-values that we always provide. And of course the numerical values anyway. And just to show you that how these plots sometimes look like, this is sample A versus sample B. And you see these clusters here. So basically here in the diagonal, that's non-differentiated microRNA. And these outliers here are particular microRNAs that are up and down regulated. And you see here multiple spots, they all represent all probes for the same microRNA. But since we have multiple probe repeats against each microRNA on the chip, you get these clusters. And yeah, I'll skip this too. It's just listing what we include. Basically, you get raw data as well as normalized and fully analyzed data. If we move on to the advantages of or features of rna-seq, of course, you get a completely different overview of your different insight into your sample. You're not really limited to just one type of molecule. Not just you don't have to have any prior knowledge of the sequence. You can look at messenger RNA, long non-coding RNA, SNO-RNA, and of course also microRNA. What's worth mentioning is that unfortunately you can't look at all of them at once from one sample using one sequencing run because the sample prep is different. If you would try to run a sample trying to sequence all these molecules at once, you would probably miss out on the vast majority of the smaller molecules, because the majority of the reads would be taken up by messenger RNA. So unfortunately, you still would have to, if you want to look at messenger RNA and microRNA from the same sample, you would have to either run 2 different arrays or still do 2 different sequencing runs. A major advantage, of course, is that it's a quantitative method, so you're getting an actual absolute number, you're getting a copy number. And it also allows you to detect structural variations such as single nucleotide mutations. Of course, all of that comes in hand with a much higher price tag than arrays. This is again just the workflow. We do the initial sample QC, in this case with a gel, do then prepare the library, do the actual sequencing, and then follow up, of course, with was probably one of the key features these days is the advanced data analysis. Just to give you some ideas what you can expect when you try to sequence a regular total RNA sample. The machine that we're using is an Illumina genome analyzer GA2X, pretty much standard sequencer these days. For the RNA-seq or whole transcriptome sequencing, we use a different machine, the HiSeq. But for the smaller microRNA molecules, this machine turns out to be a good choice. And typically you get between 20 to 30 million reads per sample. It really depends also on the sample quality. Of course, you can put multiple samples in one lane, which is called barcoding or multiplexing. You have to, of course, consider that the total number of reads doesn't change. So the number of reads that you're getting per sample decreases as you increase the number of samples. So here, for example, if you have an optimistic case or good case where you have 30 million reads, one sample, you get 30 million reads for the sample, which would be ideal for discovery work if you want to look at low abundant molecules and so on. And if you're interested in just profiling, you could use up to 3 samples per lane. That's what we would recommend, maximum 4. What this gives you is, basically in this case, 7.5 million reads per sample, which is brings you, it's in the same ballpark as the amount of data that you would get with a regular microarray. So while next-gen sequencing is really great for discovery, we at this point still feel that if you want to do profiling work, we would still recommend to use arrays, at least when you're working with well-characterized species. And the simple reason is that the sample prep in case of next-gen sequencing is rather involved, and there are several amplification steps, so there are chances that you're biasing your profile. And there's lots of discussion still going on about this and what part of the sequencing results are actually just sample library prep artifacts and which not. So if you want to really just look at differential profiling and you're working with something like human mouse rat, the array would be still our preferred method of doing it, simply because you're using the sample without any initial amplification step. So once you have the raw reads, the first thing, of course, that's being done is we try to map the reads to different sources like the genome of that species, or of course MirBase. And I don't want to go through all these steps here, but basically the different mapping steps divide up the results into 4 different groups, and of course something that's not of interest. Group 1 will be those that map immediately to MirBase, so of the known species, so these are known, group 1 are known microRNAs. And then down here you have candidate species from the microRNA genome inconsistent with MirBase, so there's a deviation from the database. Then there are known microRNAs, candidate species microRNAs, and potentially novel microRNAs, group number 4. Of course, that's the one that most people are after when doing sequencing. Just to give you a visual impression about the distribution that you can expect, in this case, out of the 30 million raw reads, 65% roughly were mappable. That's actually not too bad. Sometimes it's only 50%. Then out of these mappable reads, roughly 57% were mapped to miR-base or other microRNA candidates. For example, if we work with plant samples, of course, we can also map against the predicted sequences in the plant database, which is separate from miR-base. And then if you look at the end results, you have typically around here 70%. I shouldn't say typical because it really depends on the species, of course, but in this case you have 70% of the microRNAs that were basically confirmed with sequencing that fall into the category number 1 here. And then if you look at number 4, that's the second most prominent group here with more than 3 million reads, and 29% these are potentially new microRNAs. And if we would do a so-called seq-array experiment, we would then now go ahead and design the custom layout for the chip based on all these sequences. Besides the copy number, what you're getting, of course, is, and the actual sequences, what you're getting is a length distribution of the mappable reads. So this is oftentimes also can give you information about the developmental stage because the overall or the average length of the microRNAs changes depending on the developmental stage. So this is a typical result here with a maximum at 22 nucleotides. And probably the other most important number numbers are these here. It shows you the distribution of the so-called isoforms or isomers. So this is the part where the discussion is still ongoing about how much of this is really due to the library prep and how much is real. But what you're getting basically is let's take here the top sequence. You get 60,000 reads in this case for this particular sequence. But besides this dominant version here, you also have other forms here that are this one, for example, which is just shortened by 1 nucleotide. You still get 10,000 reads for this one. And of course, an array is not able to, a microarray is not able to distinguish between these two. So if you have discrepancies between different methods, that's probably the major reason. This, of course, also applies to qPCR. When you try to validate array data with qPCR, you certainly will not get the exact fold change, but sometimes you in a few cases you get really big deviations, and this is the likelihood that this is the reason is really high. Another reason here is you also see here some sequences that actually have some mutations, single nucleotide mutations, with a pretty high read number here. So if we move on to a couple of application examples here. If we go again through the whole, so the different steps as looking first at biomarkers and then later at therapeutical approaches. This is a paper that came out late last year. It was the first time that microRNAs were shown to be present in circulating high-density lipoproteins. And the fact that they are present in these HDLs allows them to basically being transported to and their regulatory information basically being transported to rather distant targets. And what is shown here is here on the left side is is hierarchical clustering of the samples. On the right side here, you have your normal samples. In this case, they used 6 normal samples versus 5 disease samples. And as you can see, there's clear differential expression that can be observed between the samples here. So this is basically an overview or a very basic paper which shows you the differential expression between the different stages, different types, cell types, whether it's control or normal versus the diseased one. And yeah, the main implication here was really the fact that microRNAs were found in these high-density lipoproteins and the realization that they are being transported to distant targets. So this was basically just looking at the overall profile, and then if we move on and look at a particular microRNA as a biomarker, this paper just came out earlier this year in April, I think. It revealed that microRNA 126 can be potentially used as biomarker for diabetes mellitus associated vascular complications. And what is shown here is basically the normal and diabetes samples and how the microRNA expression levels are really downregulated in the diabetes samples. What's shown here is that are the results for a couple of microRNAs, so microRNA 21, which is probably one of the most commonly found one, differentially regulated one. It plays a major role in pretty much every cancer type. Then MIR27A, 27B, so the 27th family. Then MIR20 126 was highlighted here because it shows the biggest decrease, the biggest effect, and then microRNA 130. As mentioned, it's not a good idea, or it's not realistic to expect to be able to just look at one particular microRNA as a disease marker for a particular disease. It's, as you can see, a lot of microRNAs are affected, and you have to look at an average between 15 and 20. One of the difficult aspects of these experiments is always to find the right time point to look at. And so in this study, they looked at a week-long time frame here. And as you can see, while the overall expression levels increase, the differential is still clearly prominent. Then another study that came out this year is studying basically the environmental effects, or the effects of environmental agents that are affecting the endocrine, and these were termed endocrine disrupting compounds, or EDCs. And what is shown here are basically the 3 different t-tests, how different EDCs affect the microRNA expression levels of MCF7 cells. So they were treated with different EDCs here, for example, bisphenol A, BPA, or DTT here, and a whole variety of microRNAs were found to be differentially regulated, which then again can be used potentially as biomarkers. And of course, again, MIR21 showed up as one of the key players here, as in so many other pathways. Then if you move on to the next step and look at potential therapeutical targets, this is a paper that was already published in 2010. But it was important because it showed what happens if you Well, it showed several things. First, it showed that the vascular endothelial growth factor, or VEGF, plays a crucial role in microvascular complications of diabetes. It is actually targeted by MIR93, and you can see this these graphs here. So here in A and B, there are two different the values for two different targets, and their fold changes and expression levels are shown. And you see the high glucose sample here, and then how using a microRNA-93 mimic can dramatically reduce the expression levels. And then when you reinstate VEGF, add that to the mix, then it goes back to the high glucose condition. So this was one of the first experiments where in the endocrine field that where a microRNA was shown to hold therapeutic promise. Another microRNA that shows up rather frequently and is differentially regulated in a whole variety of experiments is 146A, and this was also shown here in this case, where it contributes to where it's involved in the anaplastic thyroid carcinomas. And it was basically shown that it inhibits, or that its inhibition in FRO cells determined the block of their malignant potential. And what it does is basically it interacts with or regulates the nuclear factor NF kappa B that is strongly activated in human anaplastic thyroid carcinomas, or ATCs. And what you see here again are the samples and the FRO cells, then with an antimer, and then with an antimer 146A. And as last example, this is one from Nikolai von Oer's group at UT Southwestern. It's they looked at the thymically encoded microRNAs and showed how they are stress responsive and modulated by thymopoiesis. What they looked at again are different thymic tissue samples, one stressed and one normal condition, and the result was that in this case they used mice, and the result was that they found 18 differentially regulated microRNAs, and basically 18 that were above the threshold that they applied of a fold change of 1.5-fold. So that's a reasonable number, 18. That's exactly what you would expect for microRNA signature, between 15 and 20. So what I've shown here are just the values at different time points with PBS as a control, or with LPS as the treated version. And here the actual image scans. These are raw data, of course, and it doesn't really have, if it wouldn't be visually so appealing, it wouldn't have any, of course, meaning. Yeah, because like I said, these are just raw data, raw scans, nothing normalized or anything. But these are basically the PBS treated one and the LPS treated one. One is labeled with site 3 here, the PBS, meaning that's green, and the LPS is labeled with site 5, which is red. And the color code here in these blue chips is basically the background is 0, which is blue, and then bright red are the really high intensity, highly expressed microRNAs. And if you look at the ratio picture with the black background here, you see some that are clearly green, and then others that are more reddish. So the reddish ones are upregulated, are microRNAs that are upregulated in the LPS treated samples, whereas green are prominent in the control samples. And so while, like I said, these are just raw data, but visually very appealing, and you can immediately see what's going on. So we found that a lot of customers just started to publish these raw data. One comment about the method here. In this case, we used 2 different dyes. we don't do this any longer because so we switched to single color arrays, and we would recommend, no matter which array you use, to stick to a single color. The reason for this is simply that initially when only a few microRNAs were known, dye bias was really not an issue. There were maybe 2 or 3 out of, let's say, 700 human microRNAs that we had tested and knew that they were dye biased. And if you apply the right experimental design, you can minimize the effect of this using a dye swap model. But the more microRNAs were discovered, the more also showed some considerable dye bias. So this is when we stopped providing dual color arrays. So this as a brief overview. Like I said, I could basically touch just the surface of each paper, but I hope it gives you a good overview. And yeah, we are located in Houston, as mentioned. We have the offices in China, in Japan, India, and South Korea. But no matter where you are worldwide, all samples end up here in Houston and are being processed here, and the experiments are run here. Our European customers are typically shipping the samples directly to us, also similar to the customers here in the United States or in Canada. Yeah, I hope this was somewhat helpful to you. If you have any questions, feel free to enter them in the box now if you haven't done so.